Today we are going to learn about chapter number 16, Heredity and the Evolution. Living organisms produce new individuals that are similar to themselves by the process of the reproduction. In the previous chapter, in the chapter number 15, we have learned that according to chapter number 15, the organism na new individual reproduce by the process of the reproduction. Reproduction makalani lagi. The first type is the asexual reproduction and the second type is the sexual reproduction. So, I'll tell you a brief about what is sexual reproduction and what is asexual reproduction. Firstly, asexual reproduction male and female gametes unite tawdabado. Only the parent cell, only one parent cell na new individual or new offspring produce tawdabo kay kawi asexual reproduction. So, asexual reproduction can be defined as productions of the offspring without the involvement of the gametes. Aduga sexual reproduction sa gana male and the female gamete unite only as the result of the union. Katawi it results in the formations of the offspring or new individual. So sexual reproduction can be defined as production of the offspring or the new individual by the fusion or union of the male and female gametes. Generally, very low degree of differences are observed in the offspring produced through asexual reproduction. On the other hand, in case of the sexually reproducing organism, their offspring develop trait. Trait has the distinguished character or different character different from those of their parents. So, in case of this asexual reproduction, the offspring produced among the offspring produced there is little differences. Nasagi parents duga offspring mayam duga gi mark duga difference yam na akuna uba ngamdeba. Kam kanda asexual reproduction na produced over case da. But in case of the sexually producing organism the kadui nasagi offspring ga parents ka gi mark da kadui distinguished character or kitan character that different over character kona uba pangeba. In plants like sugarcane and the bamboo, which are generally reproduced asexually, we find a little variation. Variation is the different habdo. So, in the sugarcane and the bamboo, the parent plant as well as the offspring, what happens is that in between the parent plant and the offspring, there is very little difference. In plants like sugarcane and the bamboo, which are generally reproduce sexually, we find a little variations among the individual plant. So as long as they grow under the similar conditions. So when bamboo and the sugarcane kadoi similar condition, the same condition, the growth of matam da kadoi new organism duga parents ka gi marakta kadoi differences akoina yamna uba phangdeba. But in the number of the animals, including human being, which produce sexually, quite distinct variations are visible among the individuals. But in case of the sexual reproduction, new individual parent ki marakta differences karadi uba ngameba, except in the identical twin. DNA, deoxyribonucleic acid, also called the genetic material, provide instruction for making the structure and the functional protein. DNA the DNA is present inside the nucleus of the cell, and this DNA provide instruction people structural and the functional protein sembada. Provide instructions for making structural and the functional protein that gives member of the same species similar recognizable external and the internal features. So, due to the presence of the DNA, what happens is that there is differences in the external and the internal features of the organism, and these are called the character or the trait. A segment of the DNA molecules that provide the informations for one trait is called the gene. According to the functional segment of the DNA, ngasai information piriba, instructions piriba, segment of the DNA is called the gene. The genetic material sometimes changes or mutated due to the certain internal or the external factor. Such changes can cause the genetic material to give different instructions and lead to the different kind of the trait being formed. You know, inside the nucleus, the DNA is present, and the DNA is the genetic material. Sometimes there may be change in the genetic material due to the internal or external factor. Due to the change in the genetic material, genetic material the change it led to the formations of the different kind of the trait. Consequently, small differences or the variations in the structural and the functional character between the members of the same species are observed. Due to the change in the genetic material, DNA natra genetic material change lakpaduna kadawi akhoi structure da, akhoi body structure da or function da kadawi change laiba, even though we belong to the same group of the species. For example, in the human being also we have different structure and the different functional character, and that is due to the presence of the genetic material or the DNA. All these variations in the species do not have the equal chances of the surviving in the environment in which they find themselves. It is not necessary that all the organism or all the individual will survive in the changing environment. Depending on the nature of the variation, depending upon the nature of the differences, what will happen? Different individuals would have different kind of the advantages. Some organisms may have more advantages in living in the hot climatic conditions. Some organisms are more advantages in living in the cold climatic conditions. So, based on the nature of the variation, different organisms have different advantages in the different environmental conditions or in the different surrounding. Selections of the variant by the environmental factors form the basis of the evolutionary process, and these changes in the environmental factors form the basis of the evolutionary process. Heredity. Heredity means transfer or passing of character from one generation to another generation. Character is one generation to next generation da transfer or pass to heredity haina defined the weba. Long before the discovery of genetic materials, biologists tried to discover rules which would explain why observable characteristics of offspring are similar to those of their parents and even their grandparents. Before the discovery of the genetic material, genetic material DNA discovered biologists they tried to find out why there is similarities among some characteristic between the offspring and the parents. Parents not grandparents the similar characteristic they try to discover it. So let's take an example for the better understanding of transfer of character. For example, if the mother have curly hair, mother hair curly and the baby which is born from the mother also have the curly hair. Ngasagi mother to the pulaiba angang to sukadui curly hair oiba. That means the gene which is responsible for the curly hair. Ngasai kadui curly hair oiliba gene do kadui is transferred from the mother to the offspring or to the baby. Not only this, character can also be transferred from their grandparents to their grandchildren. That hasina habadi kadui character ma transfer to parents dagi magi daughter natraga magi offspring dadang transfer to ni have later. Character can also be transferred from their grandparents to their grandchildren. A normal child bears all the basic features of the human being. However, it shows variations from their parents. Every child have the basic features, and these basic features are all similar in all the individual. Akoi individual organism am the basic features but there is some differences, and that differences distinguish the offspring from their parent. Thus, every person possesses certain characters which distinguish themselves from the others. So, every individual, every organism has some character, and that character help in the distinguishing themselves from the other. Can you imagine how much variation exists in the human population? Here, let's see some differences in this figure. In this figure, you see the lower part of our ear is called the ear lobe. In the figure number A, in the figure number C, there is differences in the ear lobe. Here, the ear lobe is not attached to the side of our head. So, the kadui ngasay ear lobe sa kadui akoy gi head ki side ta attached to laite ba. Here, in the figure number B, the ear lobe is attached to our side of the head. This means that there is differences among these two individuals, and these small differences make one individual different from the other individual. Mendels contributions. Of the several theories that have been formulated to explain how the traits are inherited, Mendels' theory has provided the foundations upon which all the later work in the genetics have been built. So let's study more about the Mendels contributions and who is Mendel. Mendel selected the common garden pea for his experimental work. In the garden pea, he found a number of contrasting visible characters. For his experiment, Mendel selected the garden pea. Garden pea, scientific name is the Pisum sativum. So when we write in the notebook, it should be underlined separately. The genus and the species should be underlined separately. So among different type of the plant life, but why do the Mendel select the garden pea? Mendel select the garden pea because a number of the contrasting visible characters, different type of the contrasting visible characters garden pea da natraga observed. Due to that, the Mendel select the garden pea for his experiment. In the garden pea, he found a number of the contrasting visible characters, namely round and the yellow seed. In the seed also, the seed may be either round or either it may be wrinkled. Wrinkled di supado. Tall and the dwarf, wangba natraga nembado. Yellow and the green seed. The color of the seed may be either yellow or either it may be green, and so on. In the garden pea flower, the petals entirely enclose the reproductive structure, male or the stamen, which produce the pollen, and the pistil or the female, which produce the egg. So, in the garden pea, what happens is that the male and the female are found enclosed by the petals. Petals na kadui male ga female gagi reproductive structure da enclosed to thammeba. Although insect may occasionally penetrate in the flower, self-fertilization is the rule. In the garden pea, mostly self-fertilization takes place. Self-fertilization means when the unions of the male and the female take place in the same flower, that is known as the self-fertilization. In his experiment, Mendel opened the bud and removed the stamen. So, for the experiment, what do the Mendel do is, Mendel take the male reproductive structure, that is the stamen, from before they are ripened. Then, by dusting the pistil with the pollen grain from the another plant, he performed the cross pollinations between the. We have learned that in the garden pea self-fertilization take place. So what the Mendel do is that he remove the stamen from one flower, and by dusting the pollen grain to the another pistil of the another plant, he performed the cross pollination. So the law of segregation state that law of segregation has during the gamete formation, allele from each gene segregate from each other. So during the gamete formation, the gene which is present in the parent cell will be segregate. Segregate means will be separated, and these separate genes will unite together to form the new individual. Let's study about the monohybrid cross. Mendel, in order to perform the monohybrid cross, he took a character that is the height of the plant. So, in the height of the plant, we'll see two types of the plant, that is the tall and the dwarf. Since the Mendel took only one character, that is the height of the plant, this type of the cross is known as the monohybrid cross. So, this tall plant and the dwarf plant are taken as the parent, and the gene or the factor which is responsible for the tall is represented by capital T and the capital T, and the gene or the factor which is responsible for the dwarf is represented by small t and the small t, and this tall plant is taken as a male plant and the dwarf plant is the female plant. Here, one point you have to remember is that the capital T is dominant over the small t. Dominant over the chow. That means when the capital T is present, akona offspring da capital T yaura kanda kaidoni. When the capital T is present, what will happen? The plant will be always tall. Capital T yauri gaiti kaidawi. Plant cha adum tall oiniba, because the capital T is dominant over the small t. So let's see here, the tall plant and the dwarf plant is taken as the parent, and this tall plant and the dwarf plant are allowed to cross. So before crossing, the tall plant will release a gamete, that is the capital T, and the dwarf plant release the gamete or the gene which is the small t, and this capital T and the small t are fused to form the first generations of the offspring. In the first generations, you will see the capital T and the small t are crossed. As the result, we found the tall plant where you will see one capital T and the one small t. Since I have said that the capital T is dominant over the small t, the plant which is found in the F1 generations are tall. So whatever the number of the plant which is found in the F1 generation, the fangiba plants in the kadawi tall oiba, and it is a heterozygote, and it is a heterozygote plant. What is meant by heterozygote? Heterozygote had the individual having two different genes. Here, a capital T gene is present and a small T gene is present. This type of the zygote is this type of the individual is called the heterozygote, and all the individual or all the plant which is found in the F1 generations are tall and they are heterozygote individual. Now, the Mendels collected the seed from the F1 generation, and the seed are allowed for the selfing. Selfing adi kaya chap manaba ani kadawi individual ani. Habdi chap manaba di mangonda yauriba factors to natraga gene do. Adu ani chap manaba ani akoina cross to bodubu kay kawi selfing. So here, this heterozygote plant is taken as a male, and this heterozygote plant is taken as a female, and these two individuals are allowed to cross. Since two different types of the gene are present in this male and two different types of the gene are present in this female, here When the gametes are released, this individual or this male release capital T gamete and the small t gamete. Here also the female individual also release two gametes, that is the capital T gamete and the small t gamete. So in the F2 generations, what happened is we found two types of the plant, that is tree tall plant and the dwarf plant. I'll show you how these individuals are formed. This gamete of the male fuse with this gamete of the female and form one homozygote plant. Homozygote means individual having the same identical gene. Individual amada same identical gene yabudu chap manaba gene yabudu bokoni kai kawi homozygote. See here the capital T gamete of the male fuse with the capital T gamete of the female and form an individual which is a homozygote tall plant. And then next is this capital T gamete of the male fuse with the small t gamete of the female and form a plant which is heterozygote. Here capital T and the small t of the female, and it is a heterozygote plant, and this plant will be tall. Since the capital T is dominant over the small t, this plant shows the character of the tall plant. Here next also this small t of the male gamete fuse with the capital T of the female gamete and form capital T and the small t plant, which is a heterozygote tall plant. The last one is this small t gamete of the male fuse with the small t gamete of the female and form small t and the small t, which is a dwarf plant. Since there is no presence of the capital T gamete and there is only presence of the two small t gamete, the plant will be dwarf and it is a homozygote plant. So in the F2 generations, there will be formations of the two homozygote plant and the two heterozygote plant. And the phenotypic ratio, the character which can be seen with our naked eye. Character dubu phenotypic ratio. So the phenotypic ratio of the F2 generations will be 3:1, where the 3 represent the tall plant and the 1 represent the dwarf plant. And the genotypic ratio will be, genotypic ratio means when we see at the genetic constituent. Genetic constituent yengakanda, the genotypic ratio will be 1:2:1. Here this one represent this homozygote tall plant. These two represent the heterozygote tall plant and this one represent the homozygote dwarf plant. This is how the monohybrid cross is performed by the Mendel.